Inherited red blood cell disorders is caused by an atypical hemoglobin molecule called hemoglobin S, which makes the hemoglobin abnormal, causing a sickle shape or a crescent shape. Sickle cell disease is a lifelong illness, and the severity of it can change over time due to a number of other biological factors that are not yet understood. The signs and symptoms usually begin in early childhood. The symptoms are caused by a low number of blood cells, and they can include repeated infections and periodic episodes of pain. So, how does this happen? The mutation originates in chromosome 11 in the HBB gene. So the HBB gene is sort of like the recipe for the ribosome to make or synthesize the protein beta-globin. So say this is all the DNA in the HBB gene, but then this specific section of the recipe book is to make the protein beta-globin. Here's what that section looks like, unzipped. So there's 4 bases, A, T, G, C, and each of those attract another. A with T, T with A, G with C, and C with G. But here's where the mutation occurs that results in SCD. Here, where there's normally a base of T, an A base has replaced it. This mutation is called a single substitution. Now, when the enzyme RNA polymerase comes along to transcribe the DNA sequence, adding one nucleotide at a time until the message is completed, the mRNA now carries the mutation. The mRNA will now be carried out of the nucleus into the cytoplasm to find a ribosome, which is composed of ribosomal RNA, which is the shepherd you saw before. So here's where translation occurs. So every set of 3 bases is called a triplet, or a codon, as it codes for a specific amino acid. The rRNA will read each codon and act as a pointer for the appropriate tRNA. tRNA carrying its amino acid then links to the mRNA. It then drops off an amino acid, creating a polypeptide chain. This polypeptide chain, without the mutation, results in the protein beta-globin. However, with the mutation, it now creates the protein hemoglobin S, as the amino acid valine has replaced the amino acid glutamic acid in position 6. So how does this affect the hemoglobin? Well, each red blood cell contains approximately 270 million hemoglobin molecules. In the hemoglobin molecule, which is a protein in the red blood cells that carries oxygen throughout the body with the job of delivering oxygen to the cells of the body, has 4 subunits. There's 2 alpha globins, and normally there's 2 beta globins. However, in sickle cell disease, at least one of the beta-globin molecules has been replaced with hemoglobin S. There are different types of combinations that can result in different types of SCD. However, when both are replaced by hemoglobin S, this causes sickle cell anemia. This is the most common and severe, and unfortunately this is the gene that you 2 carry. So what does this mean? Well, the mutated amino acid valine causes the abnormal hemoglobin S subunit to stick or clump together. This forms long, rigid molecules that bend red blood cells into a sickle shape. So what effect does this have? Well, in a normal red blood cell, it is round and flat and flexible, and it can move through large and small blood vessels. However, sickle-shaped cells are not flexible. They stick to vessel walls, causing blockages that slow or stop the flow of blood, and therefore oxygen can't reach nearby tissues. Without oxygen, cells cannot conduct cell respiration, thus they do not produce energy, or ATP. This results in attacks of sudden, severe pain and can cause organ damage. Over time, this can cause damage to the brain, eyes, lungs, heart, bones, and skin. See, in a normal cell lifespan, they can last up to about 90 to 120 days. But because sickle cells can't shape easily, they burst or hemolyze. That means that their lifespan is usually about 10 to 20 days. See, the body can't reproduce them fast enough, meaning that the number of red blood cells are lower than normal. This means that the person now has anemia. Anemia can cause shortness of breath, fatigue, and delayed growth and development. So how does one inherit sickle cell disease? Well, it's hereditary, meaning that it's in your genes. At conception, a person receives one set of genes from the mother, in the egg, and a corresponding set of genes from the father, in the sperm. Every homology pair has one chromosome from each parent. So what is the probability of your child having sickle cell anemia? Well, each of you have only one chromosome in your 11th homology that carries the sickle cell trait. Since it is a recessive trait, this will be represented by a lowercase s. The healthy trait will be represented by a capital S. Punnett squares are used to show the probability of the possibilities of all genotypes. As we can see in the Punnett square, there are 3 different possible combinations. The first is that your child will carry 2 healthy traits. This is known as homozygous dominant. Your child will be fine as their phenotype will be healthy. They cannot pass it on to your grandchildren, and there is a 25% chance of this happening. The second possibility for your child is that they carry one of each. This means that they are a carrier. As sickle cell disease is an autosomal recessive disorder, the phenotype will still be healthy, as the sickle cell disease trait is dominated by the healthy trait. This means that they will experience no symptoms. However, they can pass it on, and there is a 50% chance of this occurring. The third possibility is that your child will have sickle cell anemia. This is because they are homozygous recessive, and as there is no dominant trait to block out the recessive trait, it means that the recessive trait shows through, which is sickle cell anemia. They will definitely pass on the sickle cell trait to their children, and there is a 25% chance of this happening. It's important to mention that the probabilities for each child exist independently of what happened with prior children. This means that you could have 4 healthy children, but you could have 4 children with sickle cell anemia. It's still a game of chance. There is also no way to alter the probabilities. But there are options. To test to see if your child has inherited the disease during pregnancy, there are 2 options. Either amniocentesis, which can be conducted 16 to 18 weeks into the pregnancy, or the chorionic villus sampling. which can be conducted in the 9th or 10th week and tends to be more accurate. As you are only 5 weeks pregnant, it is recommended that you choose the chorionic villus sampling. So chorionic villus sampling, or CVS, takes a very small amount of material from the developing placenta. This is done by placing a thin tube through the cervix to obtain the sample. It takes a sample of the cells that are of the same genetic type as the baby, and this can reveal single genetic abnormalities. Annually, approximately 5% of pregnant women are offered this. Unfortunately, there is a chance of miscarriage, but it is very unlikely. 1% of the time, it may indicate abnormality where there is none, and 1 out of 1,000 times, abnormality will not even be detected. However, the odds are still in your favour. This type of testing is still more accurate though, as more cells are actually being collected. And if your child has inherited the disease, then it's lucky that they're born now, because in 1973 the life expectancy was 14 years, but there have been many scientific advancements since then. So if they have inherited the disease, then I recommend hematopoietic stem cell transplantation. This is currently the only cure for sickle cell disease. Most transplants are performed in children, usually younger than 16. HSCT has evolved and developed to a great extent since it was first conducted. Originally, little was known about the histocompatibility antigens, and no one tried to match donors, meaning that the transplants were usually rejected. How it works now is that stem cells are taken from donors, from their bone marrow or blood. Donors can have a sickle cell trait, meaning that either of you can be donors. The recipient will then be treated with drugs that will destroy or reduce his or her own bone marrow stem cells. Donor stem cells are then injected into the recipient's vein. Injected cells will then make home in the recipient's bone marrow, gradually replacing the recipient's cells. The new stem cells will make red blood cells that don't sickle. Although the process has been refined greatly since it was first introduced, there is still a risk that the body will reject it. However, this is unlikely. If you don't like the idea of a transplant, then there is an oral medicine called hydroxyurea. It has been shown to reduce the need for blood transfusions and hospitalizations. It stimulates the production of fetal hemoglobin, which is the type of hemoglobin found in newborns that helps prevent the formation of sickle cells. However, it does include risks, as it increases the risk of infection, and long-term use might cause problems later in life. The treatment currently being developed, which will hopefully be available in the future, is gene therapy. This can be done in 2 ways. First, where they insert a normal gene into the bone marrow, or the alternative, where they turn off the defective gene while reactivating another gene responsible for the production of fetal hemoglobin. However, this will not be ready yet. Finally, if you are considering to have another child, then I recommend the pre-implantation genetic testing. It's a scientific technique which is to be used to test embryos for a specific known genetic condition, such as sickle cell disease. It enables only those unaffected by sickle cell disease to be selected for transfer during an IVF cycle, maximizing the chance of a healthy baby. This is done by karyomapping, where it specifically identifies embryos not affected by SCD. This form of testing does not harm the embryo. So to conclude, you should use the chorionic villus sampling to determine whether your child has inherited the disease, and if they have, you can terminate the pregnancy without affecting the fertility for future pregnancies. However, if you want to continue with the pregnancy, when your child is born, they should undergo a hematopoietic stem cell transplantation. Alternatively, they can use the oral medicine hydroxyurea. For future pregnancies, you should consider IVF to be able to conduct pre-implantation genetic testing.